Some quick help with transcription and translation. You need to remember that transcription happens inside the nucleus. It's very similar to DNA replication, so be careful on some of the differences. For instance, in mRNA transcription, there is no leading strand or lagging strand, since the mRNA polymerase is just reading one side. That one side, we're going to call the template strand, because the template strand is identical. It's identical to the product. This is the product here. What is the whole purpose of this process? Is to make a mature mRNA. But if you think about it, it makes sense that this mRNA being copied off of the template strand is going to be identical, except for it having U's instead of T's, to the coding strand. That's what we call the strand that is identical, hence the word coding, because this strand is is exactly the same as the product of the mRNA, which is the code that's going to be translated into a protein. So the coding strand is identical to the mRNA, except for the fact that the mRNA has U's instead of T's. Now this mRNA has to get out to where the ribosomes are, so it leaves through the nuclear pores into the cytoplasm. And in the cytoplasm, that mRNA, single-stranded, has a start and a stop. Oh, by the way, there are some changes that happen here. The process is a little more complicated than I'm showing you. There's some snippets that are cut out, introns are cut out, and they're spliced back together. But that's neither here nor there. That's a little deeper than we need to get into. The mRNA comes out into the cytoplasm, and as soon as it does, the ribosomes find this start codon, and the ribosomes act as an enzyme. They themselves are made of RNA. They're ribosomal RNA. There's actually several strands of RNA involved in making this ribosome, this organelle, one of the only non-membrane bond organelle that you've been taught, right? So when we move forward, we know that the tRNAs come in. The tRNA, now this is the key. And this is very important because when we go to translate, a lot of you have been making a mistake of using the anticodon as the code, the 3-letter triplet code to try to translate the amino acid. Each tRNA has an anticodon, complementary code to the mRNA, and this makes this process very specific, since every tRNA carries a specific amino acid. This is a degenerative code. There are duplicate tRNAs that carry more than they carry the same amino acid. For instance, there's 3 or 4 different codes for, let's say, tyrosine or alanine. But these 3 codes, as you'll see when we look at the coding again, each individual code, each individual tRNA carries only that amino acid, so it's very specific. But there are redundancies. There are more than one tRNA more than one code carries a specific amino acid. Often there are exceptions to everything, right? So for instance, the AUG's methionine is only for AUG. So there's only one tRNA that carries methionine, one of the amino acids. So as these things are loaded up or charged in the cytoplasm, they come and they find their place along this code. As they find their place inside the ribosome continues to read, and inside the ribosome, between the two tRNAs that stand next to each other inside the ribosome, there's a peptide bond that's made, and the one that gets the one to my left here, to the that I'm pointing to, is going to be kicked out without an amino acid. The amino acid stays in the chain connected by peptide bonds, making a long chain of covalently bonded amino acids, and we call it a polypeptide. Now let's go on and try to look at some coding. Okay, before we actually get into the use, how do we use the universal code to figure out which amino acid is going to go where in the primary structure? Remember, the protein's primary structure is the order of the amino acids. That's stuff that we learned back in August, that we spent so much time trying to make sure you knew. That sequence of amino acids, the primary structure, is determined by this mRNA, which was then first determined by that template strand in the DNA. That's why DNA is considered the master code, right? The book of life, if you will. So the very first thing that happens, I said, well, one of the many things that happens is these tRNAs are charged. So leucine is picked up by a tRNA that carries leucine, and you'll see which ones those are in a minute. And only that tRNA, only those tRNAs that are dedicated to leucine are actually going to pick up leucine. Histidine is picked up by GUG, or any other tRNA that carries only histidine. Inside the ribosome, as I showed you earlier, the two tRNAs find their spot. As soon as the tRNA is sitting next to the previous amino acid, a peptide bond here is formed. The empty tRNA goes off to pick up another amino acid. If this tRNA was assigned to arginine, then it would go out and look for another arginine, and once it finds it, would bring it back to a ribosome that's making proteins inside the cytoplasm. But here, this particular tRNA is waiting for the next one, which looks like it's going to be histidine, CAC. Its complementary strand is GUG. Remember, it's C goes with, and this is important. This is another issue that I see a lot of you guys making mistakes with. We learned of Chargaff's rules early on in the year, and I told you it was important that you learned it. We went over it several times in class and came back to it, and more recently in DNA replication. But remember, the rule is just simply that Chargaff found out that A and T go together and G and C go together, not any other sequence. But in RNA, because RNA can't hold the T, we find A goes with U and G with C. So we went through that when we talked about DNA replication, when we looked at some of our practice problems in class on Friday. So just remember that A goes with U and G goes with C. A goes with T and G with C in DNA, and in RNA, A goes with U and G goes with C. So when you see CAC, you would expect that C would go with G. So there's the G here in the anticodon. That A would go with U. So there's an A, and then in the codon, here's the U in the anticodon. And then C again, and again, that would be a G. So GUG matches CAC. Now, the universal code, I'm going to say it again. This is the third time or second time I've said it in the video, and many times I've said it in class. You need to understand that the codon is what the universal code is based on, not the anticodon. So don't get confused, and we'll see that when we look at the practice problems. CAC and GUG match, but CAC is the codon. That's the sequence that's on the mRNA. The sequence on the tRNA is not used in the universal code. Now, could we develop a chart that would have the anticodons on it instead of the codons? Yes. But that's not what's done. We use the codon, the triplet code on the codon, on the mRNA, and that tells us what amino acid it's calling for. Again, to remind you that DNA has this information in the form of a gene. The gene, remember, has a promoter region. For transcription, has a promoter region, the coding sequence, and then the termination sequence. That DNA can be opened up just like in DNA replication, but this is transcription. That information is available. It's available from the mRNA is made from 5' to 3', just like in DNA replication, except it's mRNA. And so it's going to read from 3' to 5'. That 3' to 5' is going to be read. That template strand is going to produce a complementary mRNA. Now, if you think about it, the two DNA strands are complementary. So if this is producing a complementary RNA strand, this sequence should be exactly the same as the coding strand, this being the template strand. That coding strand is different only in that U is present instead of T. Okay? And then finally, that RNA goes off and gets translated. Now, transcription, this is transcription, where the DNA is turned into RNA. And then translation is when that RNA is turned into protein. So the message is being copied here, transcribed here, and then here the transcription is turned into a protein. It's translated. Okay, let's go on and look at the code real quick. Now, this looks a lot like the practice problems I gave you in class. And what you need to get is that when you're going from DNA here to RNA here, only one of these two strands is going to be read. Whichever one's read. Now, the way we have positioned it here, this bottom one is the one that's going to match the mRNA. So if you have AAA, then you would expect to see here in the mRNA, you would expect to see what? Well, I hope no one said TTT. What you should be seeing is U, right? And then here, where if I give you 3 mRNAs, or 3 RNA, 3 part the codon here of UCG, then I would expect that you would know that the DNA is A, right? Because it's a T, and then here would be G, and here would be C. Now that would also mean that the coding sequence would be at the top T, C, TCG, right? So that, remember, there are T's here on the DNA, but there are no T's in the RNA. So it's a little confusing, but that's why you got practice problems. It shouldn't be too bad, though, right? So if I say AUG, and this is the beginning, right, of DNA replication of the amino acids, then you would expect that the appropriate DNA strand would be what? From the template strand. The template strand would read what? Well, hopefully you said T, A, C. So they match. They're complementary. And then, of course, the coding strand would read A, T, G. All right. So hopefully you get that idea, and keep working at it. If you don't, we'll try again down below. Make sure we get that. So if we were going to start translating, translation would start at AUG, at TAC. It wouldn't start up here, so this wouldn't be translated. So this would be no-go. This would have no amino acid there in that position. This would be our first amino acid. How would that read? What amino acid would that be? Well, you have to learn how to use both of these charts. So let's go ahead and use them. You start AUG. It's a triplet code, so there's three different letters. So A, U, and G gets you methionine. Let's try it over here on this chart. The first letter is A. The second letter is U, so it's somewhere in this column here. A and U. And G is here, and of course AUG gets you methionine. Now a lot of these charts, and I would assume that the chart that they're going to give you on the exam is not going to have the sequences there for you. I would wish they did, but they won't. So don't be expecting to see AUG. Expect to see the kinds of charts that I gave you in class, that I gave you in your practice problems. But that is to say, without these three triplet codes, they'll just say all of these are valine. I want to point something out here. You'll notice that what's cool about this chart, this is easier to use, I think, but this chart, they're both easy to use, but this is just easier. All four of these codes, what you see is that all four of these codes, each of these four different tRNAs all carry valine. So there's redundancy, right? There's backup. All four of these carry leucine. Proline's carried by all four of these. Histidine, I don't see it anywhere. So you see that each of the amino acids is carried by its proper its triplet code pairing. So I guess what's important here to understand is that you're going to be able to get there's different ways to have, there are different tRNAs that carry the same amino acid, but the amino acid is but that tRNA only carries that amino acid. So even though it's redundant, it's very specific. All right. So in any case, here you have CAT, and what you would do is you would, of course, if you want to do that, you want to find out what the coding strand is. Remember, the coding strand is identical to mRNA. It would be G, then T, then A. But now when we make the mRNA, it would have to be the G, because they have to be complementary. A would not be T, it would be U. And the T would be A. Now, assuming that there was some kind of start up here, what does the start look like? AUG, right? Assuming AUG is up here, then GUA would be, let's look at it, GUA would be what? GUA would get us valine. There's GU anything really gets us valine. And if you take a look at in this code, G is the first, U the second, and A the third, you get valine. So valine is then transcribed as the next amino acid. There's valine, BAL. If we take a look then at let's say the next code on is CAC. All right, so here we have we have CAC. We'd have CAC, which its complementary strand would be GTG, which in the mRNA would be CU. I'm sorry, not U. My bad. CUC. And now let's find that CUC. C is the first one, U is the second one, and C is the third. CUC is leucine, LEU. And it would continue. And so that's how you do this translation business. This primary structure of proteins is determined by the DNA structure, the DNA code, right? The DNA code determines the sequence of amino acids in a protein. So that sequence of the amino acids determines the folding and the shape determines the function, and the function is controlled ultimately by the DNA. That sequence, that part of the DNA that coded for something, we call it a, come on, you know this, gene. And that's what a gene is, right? It's a sequence of DNA that codes for a protein. All right, I hope this helped. Bye.